In today's lecture, we're going to look at extensions of Mendelian inheritance. During the lecture, you want to minimize all distractions by turning off music or any background noise like TV or movies, listen in a quiet space, and turn off any extra devices. Before the lecture begins, make sure that you've read the assigned chapters or reading in your textbook, and with that, you want to make sure that you've taken plenty of notes. Also, before the lecture begins, make sure that you've created for yourself an outline for the lecture to take notes. Put the slides in your own words in order to maximize your learning. All right, let's get started. Today we're going to cover extensions of Mendelian genetics. We're going to look at incomplete dominance, codominance, pleiotropy, epistasis, polygenetic inheritance, as well as environmental influences. So we're going to look at different forms of genetic dominance that are extensions of Mendelian inheritance. These are hereditary patterns that were not reported by Mendel. Some traits may be determined by two or more genes, not just a single gene, as Mendel reported when he was looking at the pea plants. Gene products may interact, and this could affect phenotype. Alternatively, multiple genes could independently affect a single trait. Not all heritable characters can be explained by the simple crosses for the traits that Mendel studied. However, in more complex patterns of inheritance, the basic principles of inheritance, such as the laws of segregation and independent assortment, apply. Patterns of inheritance of characters of a single gene may deviate from simple Mendelian patterns for the following reasons. 1. When the alleles are not completely dominant or recessive. 2. When a gene has more than two alleles. Or 3. When a single gene produces multiple phenotypes. Alleles can show different degrees of dominance and recessiveness in relation to each other. In a classic Mendelian cross, the P generation results in the F1 generation, and the F1 offspring always look like one of the two parental varieties because one allele shows complete dominance over the other allele. In such situations, the phenotype of the heterozygote and the dominant homozygous are indistinguishable. For some genes, however, neither allele is completely dominant, and the F1 hybrids have a phenotype somewhere in between those two parental varieties. This phenotype is called incomplete dominance. Another variation on the dominance relationship between alleles is called codominance. In this variation, the two alleles affect the phenotype separately in distinguishable ways. In simple dominance, if the genotype of the offspring is homozygous dominant, indicating that the offspring inherited dominant alleles from each parent, the phenotype will be that of the dominant allele. In this case, it would be purple. So just remember that simple dominant refers to a simple dominant allele and a recessive allele. Let's say you have a wild type plant cell. This cell will have two alleles for the flower color. Two uppercase P alleles indicate that the genes for purple will produce messenger RNA and ultimately the enzyme P in order to make the purple flower. Each allele will be responsible for producing 50% of the RNA, therefore resulting in 100% of the protein being produced. In this cell, you also have two alleles for the flower color, the uppercase P and the lowercase p. The uppercase P makes mRNA, and ultimately the P enzyme needed to make the flower purple. However, there's also a lowercase p. This indicates that the allele will not produce functional protein, because each allele will be responsible for producing 50% of the RNA. In this heterozygous cell, you will only have 50% of the protein for purple being produced. Because this organism is heterozygous, you will still be able to see the purple flower, even though you only have 50% of the protein purple being produced. Most simple or classic Mendelian genetic examples, this holds true, where only 50% of the protein is enough to do the job. So the majority of heterozygote individuals will still produce enough enzyme P to turn the flower purple. However, if there are two lowercase alleles, the cell will not produce functional protein. No RNA will be produced, and therefore no protein can be made, because each allele will be responsible for producing 50% of the RNA. In this homozygous cell, you have no protein being produced. If in this plant cell you have two alleles for white flower pigment, because the genes produce white alleles, which are non-functioning RNA. Therefore, the petals of this flower will be white in this homozygous individual. So before we move on, we want to look at simple dominance one more time. In most simple or classical Mendelian genetic examples, 50% of the protein is enough. And we saw some of these examples with the purple flower pigment that we just considered. So now let's look at some of the places where simple dominance does not exist. Some classical examples are the snapdragon. Again, incomplete dominance is when the offspring have a phenotype that is somewhere in between those of the parents. There are very few examples of this form of inheritance. It is rare. However, the snapdragons offer a great model system for studying incomplete dominance. To understand this phenomenon in snapdragons, let's use our plant cell example again. In particular, this plant cell, you have two alleles for red pigment because both genes produce the messenger RNA for the gene that is ultimately being produced, enzyme C. This enzyme is responsible for making the petals of the flower red. Both alleles, therefore, contribute 50% and therefore 100% in this example of the enzyme that's necessary to produce red petals is made in this particular plant cell example. In this plant cell, you have two alleles for white flower pigment. Both genes produce white alleles, which are non-functioning, so no RNA or enzyme C is produced. Therefore, the petals of this flower will be white in this homozygous individual. So in the instance of incomplete dominance, let's look at what happens in the heterozygote. This plant comes from an F1 offspring produced from one red allele carrying plant and one white allele carrying plant. In this heterozygous plant cell, you have one allele for red flower pigmentation and one non-functioning white allele because the gene for it produced the messenger RNA for the gene that is producing ultimately enzyme C is made, but only at 50%. Therefore, the plant will have 50% of the enzyme and 50% of non-functional color. This time, the petal color will blend it together between the two parental plants, the red and the white, producing a pink petal color. So in situations of incomplete dominance, there is not enough pigment produced by the enzyme in the heterozygote, where you have only one fully functional allele, CR, and one non-functional allele, CW. So you get an intermediate pigment color. So in the figure on the slide, this is again showing you the flow of incomplete dominance in a snapdragon. In red snapdragons that are crossed with white snapdragons, the F1 hybrids have pink flowers. Segregation of alleles into gametes of the F1 plant result in F2 generations with a 1:2 to 1 ratio for both genotype and phenotype. Neither allele is dominant, so rather than using upper and lowercase letters, we use the letter C with a superscript to indicate an allele for the flower color. So C superscript R for red and C superscript W for white. Next, let's consider codominance. Let's look at another complex form of inheritance known as codominance. Keep in mind that most genes that exist in any population exist in more than 2 allele forms. An example are the ABO blood groups in humans. This blood group system can produce 4 different phenotypes: AB, A, B, or O. There are 3 possible alleles: IA, IB, or I. The letter A and B refers to 2 carbohydrates that are found on the surface of red blood cells. Type A blood have carbohydrate A. Type B has carbohydrate B. AB has both, and type O has neither. Again, there are 3 different alleles: IA, IB, and I, where IA produces the enzyme that makes carbohydrate A, IB produces the enzyme that makes carbohydrate B, and I produces no enzymes. The genotypes that can be produced for individuals are either IA, or IAI. These individuals produce carbohydrate A on their surface. IB, or IBI individuals have carbohydrate B on their surface. Or IB, which will produce both of these carbohydrates on their surface. And the last combination of alleles is II, which will have no carbohydrates on their surface. Matching compatible groups is critical for safe blood transfusions. Type A blood has a carbohydrate-producing anti-B antibodies, and therefore can receive blood from individuals with type A or O blood. They can donate to A and AB individuals. B individuals have B carbohydrates on their cell surface and produce antibodies against A. They can receive blood from B and O individuals and can donate blood to B and to AB individuals. AB individuals make both carbohydrates and make no antibodies. Therefore, they are considered the universal recipient, making them able to receive blood from all blood types. However, they can only donate to AB individuals. O individuals have no carbohydrates on their red blood cell surface. They can only receive blood from other O individuals, but they can donate to all blood types, making them the universal donor. Now let's discuss pleiotropy. So far, we've examined Mendelian inheritance as though each gene affects only one phenotypic character. Most genes, however, have multiple phenotypic effects, a property called pleiotropy. In humans, for example, pleiotropic alleles are responsible for multiple symptoms associated with certain hereditary diseases such as cystic fibrosis and sickle cell anemia. So why does this happen? It's possible that the single gene can play important roles, and numerous cellular processes such as tubulin, transcription factors, RNA polymerase, RNA binding proteins, etc. 2. A single gene may be expressed in different tissue, which produces defects in both the skin and gut. And 3. A gene product may be required in early and late stages of development. Cystic fibrosis is an example of pleiotropy. A single mutation gives rise to a defect in chloride balance inside and outside the cell. So for children who inherit 2 recessive alleles for cystic fibrosis, there are several things that result. The first is an abnormally high concentration of extracellular chloride, which causes the mucus that coats certain cells to become thicker and stickier than normal. This ends up with a mucus buildup around the pancreas and the lungs, the digestive tract, and other organs, leading to multiple effects. There's also poor absorption of nutrition in the intestine, as well as chronic bronchitis and the reoccurrence of bacterial infections. Cystic fibrosis also results in male infertility. Untreated cystic fibrosis can cause death by the age of 5. Daily doses of antibiotics stop the infections. Gentle pounding on the chest helps to clear the mucus from clogged airways, and other therapeutics can prolong life. PKU is another example of pleiotropy diseases. This is a genetic disorder inherited from a person's parents due to mutations in the PH gene, which results in low levels of the enzyme phenylalanine hydroxylase. It is an autosomal recessive disorder, meaning that both copies of the gene must be mutated for the condition to develop. So some effects of PKU include changes in one's mental capability, changes in skin pigmentation, and there are other associated phenotypes. So let's move on to epistasis. In epistasis, the phenotypic expression of a gene at one locus alters that of a gene at a second locus. An example will help clarify this concept. Labrador retrievers, commonly called labs, have a dominant coat color. Black is the dominant coat color, and it's dominant to brown, which is designated by big B and small b as the two alleles for this character. For a lab to have brown fur, its genotype must be little b, little b. These dogs are called chocolate labs. But there's more to the story. A second gene determines whether or not the pigment will be deposited in the hair. The gene that determines whether or not the pigment will be deposited into the hair is big E. Big E results in the deposition of either black or brown pigment, depending on the genotype at the first locus. But if the lag is homozygous recessive for the second locus, small y, then the coat is yellow, regardless of the genotype at the brown or black locus. In this case, the gene for pigment deposition is said to be epistatic to that of the gene that encodes for the black or the brown pigment. So then what happens if we make black labs and are heterozygotes for both genes, big B, big E, little e? Although the two genes affect the same phenotypic character, the coat color, they follow the law of independent assortment. Thus, our breeding experiment represents an F1 dihybrid cross, like those that produce the 9:3 to 3:1 ratios in Mendel's experience. We can then use a Punnett square to represent the genotypes of the F2 offspring, as shown here on the slide. As a result of epistasis, the phenotypic ratio among the F2 offspring is 9 black labs to 3 chocolate labs to 4 yellow labs. Other types of epistatic interactions produce different ratios, but all are modified versions of the 9:3 to 3:1. Mendel studied characters that could be classified on an either-or basis, such as the purple flower versus the white flower color. However, there are many characters, such as human skin color and height, that are not one of the two discrete characters, but instead vary in the population in gradation among the continuum. These are called quantitative traits. Quantitative traits or quantitative characters are those that vary in the population along a continuum. Quantitative variation usually indicates polygenetic inheritance, an additive effect of 2 or more genes on a single phenotype. There are several examples of this. Skin color in humans is an example of polygenetic inheritance. If you look at the diagram on the right-hand side of the screen, you can see the difference in skin colors as it relates to various genes and which genes are expressed. There are several phenotypes. In this particular figure, they've described 6 phenotypes, ranging from light to dark skin. These different phenotypes arise from a variation of genes that are expressed. Now, as we finish up this lecture, we're going to consider environmental influences. Another departure from simple Mendelian inheritance arises when the phenotype or the character depends on the environment as well as the genotype. A single tree locked into its inherited genotype has leaves. These leaves vary in size and shape and greenness depending on their exposure to the wind and the sun. In humans, nutrition influences height. Exercise alters build. Sun tanning darkens the skin. And so all of these are examples of how the environment impacts phenotype. Performances on intelligence tests indicate that even identical twins who are genetically equal accumulate phenotypic differences as a result of their unique experiences. The phenotypic range is broad for polygenetic characters. Many factors, both genetic and environmental, can collectively influence the phenotype. Now you should have a basic understanding of Mendelian genetic dominance, as well as extensions of Mendelian genetics. These extensions of Mendelian genetics include incomplete dominance, codominance, Pleiotropy, epistasis, polygenetic inheritance, as well as environmental influences. If you have any questions about this material, feel free to email your professor or bring your questions to class or office hours. You can also reach out to the learning assistant who is assigned to this lecture course. After you finish taking the notes for this lecture, be sure to combine them with the notes that you've taken on the assigned reading. Once those notes are combined and you've had a chance to study them, quiz yourself by using the assisted reading questions, as well as the questions at the end of the chapter and at the end of each chapter section as you prepare for the quiz